So yeah, it's great to be able to share with you today some of the work that we're doing within Two Blades at the Sainsbury Laboratory. So we've touched already at this conference that we're trying to grow plants to feed a growing population, and that's becoming an incredibly difficult challenge with crop diseases. And so we lose up to 30% of crops, costing billions of dollars, and that trying to develop resistant varieties is a major challenge, not only for the lack of genetic variation that we have, but also just purely accessing and developing these into elite varieties takes a really long time, for 10 years just for the pre-breeding. And then if you want to develop a biotech trait, that will cost millions of dollars and sort of up to 16 years as well. So Two Blades is a non-profit organization that's been going for about 20 years now. And what we really do is translate the incredible discovery research that we've heard about, especially in this conference, and ensure that that makes it into the field, into products, into crops to help yield, but not only for commercial farmers, but for smallholder farmers as well. We have two research hubs at the moment, one based here in the Sainsbury Laboratory and another in the University of Minnesota, covering a wide variety of crops and pathogens as well. And working over 20 years, we've been able to curate and develop a wide-ranging network spanning both commercial partners, academic partners, and other funding agencies and non-profits to ensure that we are enabling rapid development of new innovations for public benefit. And so we've touched again upon the plant immune system, especially in the earlier session today. So just briefly, we do know that plants have an incredible immune system, and we know quite a lot about the resistance genes that are contained. And so here, just highlighting two major classes of resistance genes that we know a lot about, these so-called pattern recognition receptors on the cell surface, and these NLR immune receptors inside the cell that recognize molecules and effectors that are secreted from the pathogen and mount an effective defense response. However, we know a lot about them, but it's really difficult to actually find new functional resistance genes, characterize them, and actually develop them into products as well. So Two Blades has worked extensively across both of these resistance gene classes and beyond, and today I'll be focusing on what we're doing in particular for these NLR immune receptors within the cell. So one of our most recent projects was to find new resistance to Asian soybean rust, which is a major pathogen of soybean worldwide. And so one of our programs was to mine new resistance from Cajanus cajan for pigeon pea and transfer this into soybean. So this was done in collaboration with Corteva. And you can see here that just introducing this single immune receptor gives effective and really clear resistance in soybean. So this is now making its way into the field, which is great. And so building on quite a lot of our knowledge and partners with working within this NLR space, we discovered working with the lab at Moscow when he was at the Sainsbury Laboratory, that a lot of these known immune receptors, so these known NLRs, are actually quite highly expressed in steady state in an uninfected plant, which I haven't got time to go into this presentation now, but was actually sort of very different to what was thought within the literature. So here I'm showing you that from Arabidopsis, the model plant that we know quite a lot about, right the way through to wheat relatives, Solanum americanum, which has been used for donors of resistance in potato, and then also Cajanus cajan, the project that I just mentioned. All of these arrows here are NLR immune receptors and their expression level, and you can see that they are enriched in the more highly expressed portion of the transcriptome. And so interestingly, that gene that I just showed you that was transferred into soybean was a multi-year project spanning many resources and lots of people. If we'd taken this approach, that would have been in our top 10 candidates to look for. And so we've built this into a very effective gene mining platform that we're calling NLRseek. So we're starting with a diverse plant species and then building on NLR expression and other functional insights that we've had from our many years of working with these genes. We're able to curate these NLR collections and validate them in many different ways. So within Two Blades, we have extensive experience working in protoplast assays, also in transient and stable transgenic systems, and right the way down to looking at protein structures and building machine learning models to enhance our predictive capability. So once they've got these verified, another major bottleneck is ensuring that these are aligned with the regulations, both crop-specific and globally, so that we can actually get these into crops as well. So this is something that we excel at within Two Blades, of going right the way from discovery research, enabling then downstream product development. So just to highlight one of the new programs that we've been working on is that we started with looking at, can we find new resistance to major pathogens of wheat? So this is one of our staple crops. And again, we lose so much of wheat yield globally to many, many different pathogens. And only 2% of global germplasm collections have been mined for new genetic variation. And so we're really, major bottleneck is just finding these new resistance genes. So we started then with looking at 18 different species and mining the NLRs from these collections, and went directly to transforming them into wheat to be able to test with diverse pathogens. So we've now got a collection of just under 1,000 of these NLRs replicated in independent lines as well. And the main aim of this project was starting with stem rust, stripe rust, and leaf rust, which are present in all global wheat regions worldwide. And just a snapshot then, we've been able to clone sort of 3 times more NLRs that have been cloned in the last 30 years of resistance breeding, just in a project now that has spanned about sort of 5 or 6 years. So this has been a great success. Leaf rust and stem rust are our most advanced phenotyping programs, but we're also working then to expand across wheat blast, fusarium, head blight, and other major pathogens. So not only verifying in the planter, but we're able to then use this collection to understand more about how these NLRs are functioning and can we improve resistance as well. So for example, if we take one of our stem rust hits up here that's resistant, within the collection we have another NLR that's 99% similar at the DNA sequence level, yet is susceptible. And with the new technology that we have now for things like AlphaFold and protein modeling, we're actually able to understand where exactly in this protein the differences are, and that can really enable us to understand which regions are the most functional and the most important for binding any potential microbe effectors and things like that. So that's a very clean example, sort of 99% similar, but we also can use this collection to look at really refining the residues that are important as well. So taking our 2 resistant examples and then comparing as well to a susceptible example, we can then use that variation to really predict on the protein level which regions are most important to help us with our engineering. And so combining this with what we know as well on the pathogen side, with these effector molecules, we can model interactions and iteratively go between computational models and biological validation in the lab to really enhance our engineering. And so longer term then, we're curating a collection of NLRs that come from diverse plant species. We're able to then go into a functional insights, look at deploying these in an effective way with downstream product development for improved gene expression, and that longer term then we'll be able to almost develop de novo NLRs against any pathogen molecule. Hopefully, that's the aim. Thank you very much for listening. And so then I'd like to thank this everyone that's been involved in this very large collaborative project. So everybody at Two Blades, both here and globally as well. Our wheat transgenics were done in collaboration with Kaneka, and then especially with a team at the University of Minnesota for all the phenotyping, and then of course Matt and his team as well for continued development of this platform. Thank you.